Hi, in this lab we will be looking at the absorbent properties of UV polymerized hydrogels. Now you're probably wondering, what are UV polymerized hydrogels? Well, essentially this UV polymer is a reaction of smaller molecules called monomers and light. Light actually causes a reaction to where the monomers form crosslinks, and these crosslinks form one big polymer and often have hydrogen bonds which allow for water absorption. So in this lab we will become familiar with this UV polymerizable process and the water absorbent properties of these polymers. So today we will be making these hydrogels, and these are our chemicals that we'll be using. Vinyl acetate, which is our monomer, TMPTA, which is the crosslinker that helps the light crosslink the monomers. And this is our photoinitiator, which reacts with the light as well to form the polymer. We have polyvinyl acetate, which is the polymer of vinyl acetate, and this we'll be using as a control. And this is our sodium polyacrylate, which is actually found in diapers and is also an absorbent polymer. So we'll be looking at the difference between our UV cured hydrogel and then an already commercially produced polymer. So essentially we will be making our polymer and putting it and UV curing it in a petri dish, and then we will take it out of the petri dish and put it in an already weighed tub of water. So once we take out the polymer or the hydrogel, we will be able to see that there is less water in the tub, and the change in water mass is how much the polymer absorbs water by its crosslinks and the hydrogen bonding that it is able to do. Alright, let's get started. One quick safety note: UV light can be detrimental to your corneas and other parts of your body, so make sure to always wear your UV glasses when putting the polymer or the monomer solution under the UV light, and always wear gloves when handling chemicals.
To begin with, weigh out 10 grams of the monomer, 1 gram of the crosslinker, and 1 gram of the photoinitiator. Then repeat using different amounts of crosslinker. Mix the measured chemicals in glass vials. Then place the glass vials of the different samples under the UV lamp and wait for polymerization to occur. Make sure you are wearing your UV glasses. You will see the solution harden as the monomer reacts with the UV light to form the polymer. Carefully break the vial, remove the polymer, and place it in a beaker. Pour 100 milliliters of water over the hydrogel. Let it sit for 10 minutes in order for it to absorb the water. Then pour the water from the beaker into a graduated cylinder. Record the volume and subtract it from 100 milliliters. This is the amount of water absorbed by the hydrogel. Compare these measured volumes to the controls, the commercially available diaper polymer, and to the commercially synthesized polymer that we just made. Our findings show that there is an optimal amount of crosslinker for water absorption. That is 10 grams of monomer to 2.5 grams of crosslinker. This allows enough crosslinks to form a polymer that has enough space to allow hydrogen bonds.